Hello, I'm Alan Landay at Rush University Medical Center in Chicago. I'd like to welcome you all back today to our second day of the HIV and microbiome program. We're very excited by our program today, and I think you all hear some very novel and new data in the field of the microbiome. On behalf of my other co-chairs, Dr. Ronald Coleman, who you also see on screen today from the University of Pennsylvania, and Dr. Grace Aldrovandi from the University of California, Los Angeles, I'd like to welcome you all to today's program. To get involved in the program today, we really would like you to ask questions and submit your questions through the live Q&A with our speakers today. This is very important to get good interaction since we're not in person. This is really the way of getting good interactions between the speaker and our audience. We'd also ask you to please view the posters on the poster gallery today. You also have a great opportunity to chat and connect with people through the meeting hub. Gives you a great opportunity with all the participants from around the world. We have up to 13 different countries represented. And in this virtual format, it really gives you a great opportunity to have a global view of what's going on in the microbiome and HIV field. And then finally, if you have any technical issues, you can click on the live support hub. Your feedback is important. Please fill out the session surveys. This will help us next year, as we hope we'll be going live back to our venue at the NIH, and we really like to have your evaluations after the event to use it to improve the program for next year's meeting. It is really my great pleasure to introduce our keynote speaker for today, Dr. Cynthia Sears. Dr. Sears is a professor of medicine, oncology, and molecular biology and immunology at the Johns Hopkins University School of Medicine and the Bloomberg School of Public Health. She is the microbiome program leader of the Bloomberg Kimmel Institute for Cancer Immunotherapy at Hopkins and director of the Johns Hopkins Germ-Free Murine Facility and co-director of the Hopkins Microbiome Forum. Her background is as an infectious disease expert, focusing on enteric infections for her whole career, and is studying the pathogenesis of enterotoxigenic bacteria, the Bacteroides fragilis, both in the lab and clinical setting. This work has gone on over the past 25 years. The focus of her laboratory is to determine how the microbiota and specific bacteria contribute to carcinogenesis, especially that of colon cancer, and that will be the topic of her presentation today. She's also looking at the responses of the microbiome in cancer immunotherapy. She is using both human and mouse models and exploring the areas of microbiology, bioinformatics, and immunologic methods. Dr. Sears has also served as an associate editor of Clinical Infectious Disease from 2000 to 2016. She's an active member of the Infectious Disease Society of America, IDSA, for more than 20 years, and she's a current past president of IDSA. It is really a great pleasure to introduce Dr. Sears today to give this keynote lecture, and she'll be discussing and talking about probing the microbiome and disease from CRC to HIV. I think we've left off the V there, but Dr. Sears, thank you so much for doing this.
Good morning, everyone. I want to thank the organizers for giving me the opportunity to speak today. My disclosures are here related to research grants. And the goal of my talk today is to present approaches that may assist in defining microbiome interactions with a disease or condition. Since we've worked predominantly on colon cancer, I'm going to start there, and then at the end just show you a little bit of data from HIV. So my laboratory works in 3 areas. I'm going to focus on the top 2 today. In one case, it's how individual organisms can contribute to a disease process, and the second is how communities of organisms may contribute to disease. All the work I'm going to show is translational, involving both human samples and preclinical murine models. And I think it's safe to say, and you would all agree, that microbiome work is interdisciplinary, a true example of team science requiring a multiplicity of approaches. And with that in mind, I use this slide to illustrate why I don't think we're going to be sequencing our way to translation, to bring something to the patient's bedside. And I use an example, these two outstanding papers in Nature Medicine that have many other aspects that are important. They looked at over 900 fecal metagenomes, or shotgun sequencing between them. And the key point is that in both papers, when they look carefully at the predictive power of the data for colon cancer, it fell just over about 80%. And adenomas were either not included or had a much lower predictive signature. And the point is that you can't miss cancer. So if someone had a positive test, as discussed here, they needed a colonoscopy. And if they have a negative test, they need a colonoscopy. So in this case, this predictive value fell short of what is needed to take care of patients. The reason we study colon cancer is twofold. One, we are in the midst of an emerging epidemic of early onset colorectal cancer in individuals under the age of 50, dating back really as far as 1970s cohort. This is a feature of health disparities, and shown here are two individuals, both with stage 3 colon cancer, and as we know, Chadwick Boseman lost his life recently to this disease. This is a global problem, so it's, in my view, an important problem across the globe, even in areas of the world such as Africa, where we expect colon cancer to be less frequent. So the overarching goal of our work is to try to corral the microbiome to inform prevention and therapy in human colon cancer. So this slide illustrates the idea of single microbes can be important. I want to thank Dr. Xiaoguan Wu and Franck Cousseau for their work, and I'm going to focus on a bug we've long studied called enterotoxigenic Bacteroides fragilis. Humans are commonly colonized with both Bacteroides fragilis and the strains that make a toxin. And what we've learned over time is that this toxin is key to pathogenesis. If you take that gene out of the genome of the bacterium, all the tumor effects and signaling effects go away. We understand the toxin can induce DNA damage. It triggers pathways in the host that are linked to colon carcinogenesis. And in fact, this can happen very quickly in a mouse within a week of colonization. Of course, then the tumors grow. Now, this isn't the only example in colon cancer. There are other organisms, PKS E. coli and Fusobacterium, they're important, but it's an example of how one bug can teach you a lot about a disease process. Now I'm going to talk a little bit about community as a driver of colon cancer pathogenesis. And here I'm going to talk about biofilms, which are invasive aggregates of bacteria that directly interact with the colonic epithelium and are common in colon cancer. So by that, this cartoon illustrates what I mean. The entire colon is covered with a dense mucus layer and a loosely organized mucus layer. And that loosely organized mucus layer can be invaded by bacteria shown on the normal side of this, but a biofilm forms when that dense inner mucus layer, which is normally nearly sterile, is invaded by bacteria. So the pictures on the right illustrate binding of fluorescently labeled probes that illustrate these bacterial populations. So in the healthy colon here, you can see the mucus layer in light blue. But when it's invaded here by a polymicrobial biofilm and here by a biofilm that's predominantly in green, Bacteroides, and in yellow, Fusobacterium, these are striking structures on the mucosa. We've been able to show that these come sort of in 3 flavors. The majority are polymicrobial, and then there's a large subset that actually have invasion with Fusobacterium, and a much smaller subset that appear to be proteobacteria, gram-negative rod dominant. And when you look at colon tumors, you can see these are particularly prevalent on the right side of the colon in both US and Malaysia cohorts. And in Malaysia in particular, these are common in the left colon for reasons we don't fully understand. However, when you look at individuals undergoing screening colonoscopy, about 15%, whether on the right or the left colon, display these biofilms. Now, does it matter? Does this change the function of the tissue? And I don't have time to show all the data, but the red starred areas are steps that we've been able to identify. So I just showed you biofilm formation. And when we look carefully, and here's a picture of this, using immunofluorescence staining, we can see that there's a change, a likely change in barrier function because there's decreased E-cadherin, a critical intercellular protein in the colon. So here on the left is the biofilm positive sample, and on the right is the biofilm negative. On the top are colon cancer patients, and on the bottom are screening colonoscopy patients. And you can see in the normal condition, whether in colon cancer or screening colonoscopy, there's a lot of E-cadherin staining, and this is markedly diminished in the setting of a biofilm, strongly suggesting a decrease in barrier function. In association with this, we see immune activation, we see phosphorylation of STAT3, and we see the precursor to tumors, colonic epithelial cell proliferation. This is shown here. Again, these are colonoscopy biopsies, and this is IHC, Ki67, which is a marker for cell proliferation. And you can see there are fewer brown cells in the biofilm negative sample than in the biofilm positive. And all these results can be quantitated and show the same results. We were also working with Gary Zustak's lab. We were able to show that there's a unique polyamine metabolite that is associated with biofilms, and this is N1N12 diacetylspermine. However, this doesn't tell you there's carcinogenesis. So really the next question was, are these biofilms carcinogenic? And to answer this question, we teamed up with Christian Jobin's lab at the University of Florida and work done by Sarah Tomkovich and Christine De Gea. And we looked at a slurry made from biofilm positive patients that we then put in germ-free mice that are susceptible to colon tumors. And what we were able to identify is that all the biofilm positive samples, which here are the tumors, biopsies, or the normal flanking from the tumor patient, led to induction of tumors in the colons of the mice, but that were not seen when the mice were inoculated with biofilm negative biopsies. Dr. Julia Drews in the lab has taken this a step further, now taking the examination of this to individual patient samples as shown here. And what we've learned is not every biofilm will induce tumors in the colon of a mice. However, some do, whether they are biofilm positive or biofilm negative, as shown in this slide. In green is the biofilm negative, and blue is the biofilm positive, and here is the control, sham, as well as here is the control of the slurry that was initially examined. She has now been able to culture bacterial isolates out of this particular sample, 3728T, and using these 30 isolates, as shown in this graph, she can actually recapitulate the tumorigenesis from the slurry. So this gives us a microbiology result and a way to try to understand exactly which organisms and their interactions may be pro-carcinogenic in the colon. I want to mention one more thing about biofilms, and this was sort of an accidental observation. In the times we were collecting colons and examining biofilms, Christine De Gea was fortunate to get a few samples from individuals with a rare hereditary syndrome for colon cancer called familial adenomatous polyposis. And what, in this case, patients were getting a colectomy to protect them against colon cancer. And we learned a couple of things. First, the staining was very different than what we saw in sporadic colon cancer. And in fact, the majority of the bugs on the surface of the colon were either an E. coli or a Bacteroides fragilis. And second, these biofilms in red dots actually went all along the axis of the colon. suggesting that once a biofilm forms, it may be quite extensive in the colon. And they were seen on adenomas or on the normal tissue. Additional studies traced these back to the PKS positive E. coli I mentioned and the enterotoxigenic Bacteroides fragilis. And in studying those organisms' interaction more, we identified that those virulence factors interact and are synergistic in their action in tumor formation. So there's quite a bit that can be learned through microbiology, virulence factors, and different disease models. Now, microbiome research, you might say, has been dominated by big data, as I mentioned up front with the metagenomes that I mentioned. There are a variety of big types of data that can be used to address microbiome research and to try to derive both microbial and host signals. So one possibility is databases. These are large data, listed here are some large databases that have been used. Then there are sequencing methods, and this, the most commonly used still is probably 16S RNA amplicon sequencing that just detects bacteria. But metagenomics, which is shotgun sequencing, which allows you to assemble the DNA and can allow you to pick out both microbes and host responses, is another possibility. As well as bulk or single cell RNA-seq, again, can be related to either microbes or to the host, and TCR-seq if you want to drill down to exactly what's happening in the immune system. I'll also mention a couple others: computational meta-analysis, which I'm going to introduce in a moment, metabolomics, which some think are the final common denominator in microbiome research, proteomics, still in development, and ultimately machine learning and artificial intelligence, I think we will see applied. So I'm going to give you two examples here. This is the work of Jiajia Zhang, and we had the opportunity to get data from the Clinical Practice Research Data Link, which is the largest electronic health record in the world and housed in the United Kingdom. And with the help of their research team, we were able to set up a case control study matched by year of birth, gender, practice site, and the registration year. And these data stretch from 1989 to 2012. And what we were trying to look at was the relationship between oral antibiotic use and the subsequent incidence of colon and rectal cancers. And so the basic results are shown here, and there were a couple surprises. So in the colon and on the x-axis is the total exposure to oral antibiotics, and on the y-axis is the onset or the detection of colon cancers in association with that antibiotic use. And what you can see, with as little as 15 days of antibiotic exposure, there was an uptick in the detection of colon cancer. In contrast, in the rectum, there was a decrease in association with oral antibiotic use, but it was slower and it plateaued at about 60 days. When we looked at more detail at the colon, all of the signal came from the proximal colon, which is where the oral antibiotic concentration will end up being the highest in the colon, and there was no signal in the distal colon. One aspect of this data that made it, if you will, in my mind, more believable was that the antibiotic exposure association with the onset of colon cancer was more than 10 years prior to the diagnosis. So it was not near the time, which might have been a confounding effect of the onset of the cancer, but really distant from the cancer with a subsequent onset. The other method I want to mention is what we call a computational meta-analysis. And in this, a key feature is that we're reanalyzing the raw sequence data, not the published results as in a typical meta-analysis. And in our case, we use a high-resolution pipeline called Respira Insight. And the reason we use that is it has a higher species-level assignment than some other commonly used tools. So in this case, in colon cancer, we looked initially at 11 cohorts, nearly 500 colon cancer patients, and we're able to identify a consortium of 5 organisms consistently enhanced in colorectal cancer, whether looking at healthy or at normal tissues in a cancer patient. And then we were able to find identical results in our colon cancer cohorts as a validation cohort. And it's exactly that approach, working with Sue Tunham and Adeline Koa and James White, that we took to address, to take a look at how the HIV microbiome looked across studies. So the key points of this study is, this is from the first papers we found, which were in 2013 until the end of 2017. And at that time, we came across 22 studies with 17 available data sets. We again looked at 16S rRNA amplicon data. Now this represented over 1,000 samples, 300 HIV negative and about 700 HIV positive. And I also want to give great credit to all the authors of these papers who all worked with us to get the data sets right for this analysis. We were able to publish our first paper looking just at alpha diversity across these data sets in clinical infectious diseases last year. And what we learned was in the stratified analysis in which we controlled for HIV gender and sexual orientation, we identified a decreased alpha diversity only in women and men who have sex with women, not men who have sex with men and persons living with HIV. And the overall results suggested that HIV gender and sexual risk categories should impact alpha diversity and should be considered in analyses. We're in the process of taking these same data and now trying to look at the taxa associated with HIV and sexual orientation. And our preliminary results suggest that MSM status has a stronger impact on the gut microbiota than HIV status. One of the issues in across the microbiome field has been many small studies getting published with limited clinical metadata, and I think this is an area where the entire microbiome field perhaps can do better over time. I just want to mention 2 challenges in the field. One is that there can be marked technical differences between labs and between papers. And this is emphasized by the Microbiome Quality Control Project led by Curtis Huttenhauer, looking at differences amongst 15 labs. And the second thing I want to emphasize is how we probably need to think more broadly about the exposome. There appear, as we all know, many factors that impact the microbiome beyond diet and medications. Genetics and epigenetics probably matter, as well as disease interactions. So this is something in collection of metadata that we certainly need to consider. And lastly, I just want to thank everyone in the lab, our collaborators at Hopkins, our collaborators outside of Hopkins, and all of our funders, which has included CFAR for the HIV work. Thank you very much.
Well, thank you, Dr. Sears, for this really outstanding talk. And it was really enlightening and gave us a lot of new thoughts in terms of the intersections of HIV and cancer. I do have a few questions. Maybe I'll ask one before we go to the chat room, and that being, you talked at the end about your meta-analysis of data on the HIV cohorts, which I think is great. Have you been able to dig any deeper to see about the link with cancer in those cohorts, especially colon cancer, which has been your main area? What's that relationship intersection between HIV and colon cancer directly?
Again, I want to thank the organizers for giving me the opportunity to be here today. That is a great suggestion. We have not done that. In this particular data set, there was no linked cancer metadata, but it's a great suggestion. There may be enough data out there now to try to take that on. My understanding is that so far colon cancer has not had a strong signal with HIV, but there was data saying that persons with HIV get more colonoscopies, maybe because of some GI symptoms. So you may have that positive effect of prevention strategies happening.
Right. And I think that it's a great opportunity to link it with, as we're so interested in the comorbidities associated with aging and HIV, of linking those two together. Let me go to the chat room. And Dr. Alessandra Borgogni, did you analyze the presence of CRC associated polymicrobial microfilm and metastasis? And she and thanks you very much for an excellent presentation.
Yeah, thank you for that question. So, the background, I think, of that question is a beautiful paper by Susan Bullman in Science now about 2 years ago, where she demonstrated that fusobacterium went off to the metastases and actually had a consortium of additional organisms, including Bacteroides, in those metastases. We, however, have not had the opportunity to do that, so I don't really have any specific comments about linkage there, but I would refer you to that work.
Great. And then Dr. Christina Somas, again, thanks for an outstanding presentation. You also have data from the UK cohort about simultaneous use of probiotics in those individuals.
So again, a great question, probiotics being a tough topic in the microbiome field. However, in that cohort, that data was not available to us, but it's a great question.
Great. And now from my co-chair, Dr. Coleman, again, great talk. Is there anything in the HIV meta-analysis data to see risk of cancer change, which I think is kind of what you were answering earlier as well?
Yes, I from the data I don't think so. We had pretty limited data on viral load, CD4 counts, but we didn't really have clinical outcome or progression data.
Okay. And looking at the cancer side, what about class of antibiotics in your data sets? What does that contribute?
Yeah, so very interestingly, the proximal colon effect drilled down to penicillin. And actually, when we took a subset of the data and looked at people exposed to no antibiotics versus penicillin alone in the data set, and when you have nearly 30,000 cancers and 137,000 controls, you can accomplish that. This relationship held up in the data. Now, it was completely different for the rectum, where we saw a decrease in incidence of rectal cancer with oral antibiotic exposure. In that case, it drilled down to tetracyclines, interestingly.
And then from Della Pinto, thank you, regarding antibiotic exposure. Oh yeah, how long was the type of exposure? I think you did talk a little bit about that.
Yeah, I realize those graphs are a little hard to grasp in a second. So what we looked at was total antibiotic exposure over the time, from the time the individual was enrolled in the UK cohort until the end of the data, which was in 2012. So those were total days of exposure. And if you looked at it as episodes of exposure, it didn't really change the data. And it was a total surprise that such a short exposure, really as little as a single course, changed the likely incidence of the association with an increase in incidence. I want to be very careful. These are associations. These are not causal findings. This was a case control study, and so there's much more work to do. But there is data from, for example, David Relman's lab, and actually a number of labs now, that show that that impact of oral antibiotics on the gut microbiome isn't as simple as it fixes itself in a few months. There's probably, in the way I view it, is we probably accrue our own personal level of dysbiosis with some modest changes in the microbiome that may have functional impact. And I think we don't understand that as yet.
That's great. You mentioned metabolomics as a key area. What do we know? You talked about polyamines a bit. I think that's a critical area because in much of our discussion yesterday, it was about who's not there, who it's not about who's there, it's about what are they doing. And I think that's a critical part of the microbiome field. Cindy, and can you tell us a little bit more about that in the cancer studies?
So, great question. And I think we're going to see this particular area balloon over the next 5 to 10 years. So, in the areas I'm involved in with cancer, some of the highlighted metabolites so far have been short chain fatty acids with some differing results depending on which checkpoint inhibitor you get. And in the general, the microbiome and short chain fatty acids is an extremely important topic for function. The second group that has recently come to light, and just a paper in Science a few weeks ago, is inosine, where again, there's debated um, ideas about how those metabolites may impact function in the microbiota and in the host. And the other one I'll mention is something called gallic acid, which I must admit I know very little about, but there was a recent paper in Nature linking this to function of APC mutations in a sort of a very interesting set of analyses. Uh, I think this is scraping the surface, and I don't actually believe my knowledge is perfect on this topic.
Um, Laurel Langlois has a question: butyric and butyrate. That's important in gut health in terms of maintaining gut barrier integrity and enterocyte function. Do you know anything about butyrate producing bacteria in cancer?
So butyrate is one of the short chain fatty acids, and it's extremely important for the health of the epithelial cells. I don't know specifically. I can tell you in our data set, the signal was from propionate, and so we're about to launch into looking at this in more detail. But I think it's an extremely important question. There could be differences amongst the short chain fatty acids in their impact and function.
And again, a lot of what drives the short chain is the kind of diet and approaches. So in either the human studies, let's say from the UK, is there any diet history in the UK biobank? That's a hugely rich resource on which you looked at antibiotic. But what about diet in those same individuals? And I think it's fascinating that you saw this 10 years preceding the development then of the cancers in those patients.
Yeah, that is, the UK database is pretty amazing. It's all these primary care practices throughout Ireland, Scotland, and the UK. And so they've really engaged primary care physicians in entering data that is pretty expansive. But one of the things that isn't there is diet. And as I think everyone knows, capturing quickly what a diet is and how it may impact the microbiome is really a tough question. It's a tough question.
Yeah, I know the group, Paul O'Toole's group in Cork, is a lot of his work was in the aging microbiome field, really only saw the results of his work showing things in terms of different elderly populations, was really when he dove down into the diet with Fergus Shanahan and his work together with Paul. So I think it is critical, and I think it's difficult though in many of these studies to gain those insights.
Correct.
Um, are there other opportunities to think about linking HIV and cancer and microbiome? What are your thoughts about the future? Because many things that we're looking at is, we've, as with a lot of these areas, we had the Human Microbiome Project over a number of years. What do you see as the future of microbiome work? Because we're still sort of searching for the holy grail of, as you mentioned, interventions, and are we going to be able to use these microbes as diagnostics for cancer? If you were advising a young investigator today, let's say somebody in your group, what would you say they should be thinking and looking at as a young investigator?
Well, I'm doing just that with Dr. Faiza Shaikh, who just joined the faculty and who's very involved in our microbiome and immunotherapy project. And the strategy we're going to take is pivoting a bit and focusing on metabolites and then working back to the structure of the microbiome, or at least that's what we hope. integrating mouse models to see if we can drive to mechanisms, recognizing that these transitions from human to mice are imperfect at best. So I think you have to be careful. And then we're just going to be practical, in my view. We're going to try to do what I consider just small human trials to collect information, doing things that most of us would view as not dangerous to the patient, such as studying fiber in short-term studies, which of course is very much related to short-chain fatty acid production. Yogurt is another common probiotic, and there's a variety of nutritional literature that suggests that simply eating yogurt every day might be a good thing for us. Now, all of that data, again, is association, but the harm from yogurt would be, in my mind, zero, probably, and a way to try to get at some of these functional aspects in small groups of patients. So we're going to try a pragmatic strategy to step forward.
Wonderful. Well, thank you again, Dr. Sears, for an absolutely outstanding talk and great discussion from the audience. And we'll take a 5-minute break, and everybody, please come back to the next session. Thank you again.
Thank you, everyone.